Thanks, Ben, and thanks to the organizers for the opportunity to present my group's talk. So we are the Complex Networks Lab, or in other words, we do network science. These are my PhD students and my undergrads, and things have quite changed since the picture was taken about a year ago. Boy is currently a PhD student at Stanford. Will is going to start her PhD, or actually graduate studies. I'm not sure yet if master's or PhD program at MIT. Jen is going to Silicon Valley, and I hired 4 new undergrads because I've had fantastic experience publishing with them. They're just very, very ambitious kids. Of the grad students, Lei and Fazl and Yuri are all going to be done in next 2 months, and they're going to Google, Microsoft, and Facebook. So despite us doing bioresearch, social networking companies seem to be very interested in what we do. These are the funding agencies, and most of the work that fund my group, and most of the work I'll be talking about today has been funded by NSF. We study networks. We do anything and everything with networks in many domains, but most of the computational problems we are dealing with are motivated by problems in biodomain, and we study these hairballs, meaning any omics data that can be modeled as a network, or integrating multiple omics data into a network, and studying the structure of the network, the patterns of interconnectivities, and trying from this to learn something about cellular functioning. And one of the cellular processes we are very interested in is to study human aging. There's 3 reasons. Number 1, we all want to live longer, happier, healthier lives. Number 2, NSF will not fund disease, but they will fund aging. As a computational scientist, that's very important to get funding from NSF. NIH funds both study of aging and disease, as many other things. And 3, it's a dynamic biological process which enables me to model cell as a dynamical system and then do dynamic network analysis, a computational problem I'm personally very interested in. So, what do we currently know about human aging? Gen age, I don't know how familiar you are with aging, and I was until maybe 5 years ago, other than we all do age. Gen age contains about 260 genes in human that have been implicated in aging. Of those, 3 have been implicated in aging experimentally. The rest come from computational analysis. Reason is why we can study human aging experimentally. We live a very long time. And there's lots of ethical constraints. So what people typically do, these are all computational and statistical analysis. Many of you might not think of genomic sequence alignment as a computational strategy. It is nothing but computational prediction. So 2 typical strategies are collect expression data in human at different ages. These measurements typically come from different individuals. And then if a gene's expression significantly changes with age, the gene is implicated to be aging-related. Aging-related can mean 2 things: promotes longevity, which is very good, or causes premature aging, which is what we want to avoid. So being implicated in aging is not necessarily a bad thing. Second, model species such as yeast, fruit fly, worm. In those species, you can study aging experimentally for lack of those 2 key constraints. And then you can transfer knowledge from an experimentally determined aging-related gene in model species to a sequence similar gene in human through genomic sequence alignment. What's the key drawback of both of these types of approaches? There's very many approaches within each of these. I'm just giving you a high-level idea how this is done. But the key disadvantage is they're all ignoring interconnectivities between genes, proteins, gene products. So for the purpose of my talk, I will be using terminology. Gene and protein is the same thing. Biologists, please don't hate me. So 2 key ideas we've been working on in the context of studying aging is, let's pretend we're in a perfect world where we have biological networks at different ages, just as we can measure gene expression data. We don't have such experimental data due to limitations of biotechnologies, but we have static network data and dynamic gene expression data. Maybe we are lucky that for some processes we have protein abundance data, which we are still not there yet. But can we combine the 2 data types to infer these age-specific networks? And then, analogous to what is done from gene expression data alone, from the combined network and gene expression data, can we predict as aging-related those genes whose network positions significantly change with age? Second, can we redefine the notion of sequence homology to network homology through the notion of network alignment by trying to find, we've heard about network alignment from Ben, but trying to find network similar regions between molecular networks of different species, so align them properly rather than misaligning the edges? And if so, can we analogously transfer experimental aging-related knowledge from model species to human between conserved network rather than sequence regions? We've done lots of efforts on both. In my talk, I'll primarily focus on my pointer. Give me 1 second, please. I will, in my talk, primarily focus, technical issues, I apologize, on the topic of dynamic network analysis in the interest of time. My goal is to tell you what we do, why we do it, and give you a conceptual idea, high-level overview, how we do it. There will be a zillion questions, I'm sure. You will find lots of drawbacks, I'm sure. Feel free to ask me throughout the talk or after during the Q&A. And I will briefly maybe devote 1 minute if we have time to this topic, just giving you some keywords and pointing you to some of the references if this interests you, so you can check it out on your own. So if this is the current static, of course it's a toy example, or static representation of the cell, whatever data we currently have, well, maybe not all genes are active. I'll tell you a little bit what it means. Super expressed, relevant for the given time, age, condition, tissue of interest. Maybe we are only interested in the subset of those genes and some subset or all of the interactions between those genes. This is kind of relevant to dysregulated pathways if you really want to think about it, but we are more interested in the temporal aspect of inferring networks corresponding to different times and then studying evolution of network structure from global, local, whichever perspective you would like to do. Now, in the real life, we don't have such data. But we've heard a lot in this particular workshop by people who are actually in the audience, a lot about data integration, as well as it's been recognized that dynamic network analysis is needed to better model cellular functioning. And in particular, the two are not distinct ideas. They're actually quite related, the networking integration to achieve network dynamics. So what we do is, so let me try to see what I can do with my laser pointer. The laser is working, it's the slide changer, it doesn't, so it's causing me some issues. Okay. So what we do to first infer these networks, and that was not the key concept, the key goal of our project. The goal was just to use a proof of concept strategy to do so. But what we do to infer the networks, and then what we do to analyze them, to study human aging and deepen our knowledge about human aging compared to traditional gene expression and sequence-based analysis. Okay, so we are dealing with a static protein-protein interaction networks. We are using PPI data is a proof of concept. You can feel free to replace with any other type of data that you would like. We base our study on HPRD data as well as BioGrid data separately to make sure our approach is robust to the choice of the PPI network data. We then combine the static network with aging-related gene expression data. In this case, we were first dealing with microarray data, and then reviewers suggested, why not try RNA-seq? Maybe you get different predictions, maybe you get similar ones. We actually have a significant overlap between predictions that we get independent of which of these two data sets we use. Majority of the results I will report are for using this particular gene expression data. We have 37 ages, multiple individuals per age, and multiple samples per individual. And this data allows us to compute for each age, for each gene, its detection p-value, which is related to the overall abundance of its mRNA in the given age. And we have a way to, based on these p-values, keep only genes that are active in a given age and keep interactions between those genes. So initially, we kept all of the interactions between the genes in the network. Later on, we had a follow-up study where we removed some of the non-redundant, irrelevant non-central, non-important, however you want to call it, edges. I'm not going to go into details there. If you would like to know more, please follow up with me. Both strategies produce meaningful predictions, and actually removing some of the interactions to make the network sparser, so getting rid of the irrelevant edges helps the process of network inference. And here's just an illustration of 37 networks that have between this many nodes and this many interactions.
In our hands, at least, removing proteins because they are not expressed was less powerful than reweighting the edges. So have you tried that, to reweight the edges based on the expression rather than simply removing?
We have not combined in the process of inferring edges expression on top of the PPI data. Lots of ideas we've heard here through the process of network propagation or diffusion you can use for that purpose, and that's definitely something we are exploring. But we are very interested not just to do propagation here and propagation here and propagation here. I'm using propagation as just, again, proof of concept. We are very interested to consider interactions between these different networks at different ages somehow, and we have no idea yet how. So that's definitely currently funded idea. Again, this is just the proof of concept. And for applied scientists who typically use arbitrary network inference method, in particular, this study focuses on constructing gene co-expression networks and such as Pearson correlation and arbitrary cutoff of what an edge is versus is not. We have a study that says how the choice of the network inference method directly affects what biological question or questions you might be able to answer with the resulting networks. So this is definitely a valid point, and I doubt that there will be network inference approach that will work for every possible application the best. So you definitely want to target whatever question you're asking.
For each age, you are only keeping the genes that are specifically expressed at that age.
So the question is, do we at each age keep genes that are specifically expressed at that age? Can you define what you mean by specifically?
I'm asking you, how do you define?
Okay, so in a given age, we compute the detection p-value of each gene at that age. It's computed as intensity value of the perfect match probe of the given gene has to be significantly higher than the intensity value of the mismatch probe for the given gene.
With other ages?
No, it's within a sample. This is a widely adopted strategy, especially by the authors of the paper whose gene expression data we used. I'm not qualified to make such judgments on my own. So we used, for both microarray and RNA-seq data, two different strategies. We used how to define what an active gene is, guided by the authors of the paper.
What is the tissue?
I'm sorry? This is for brain tissue, because this gene expression data, and both are for brain tissue, probably it's typically studied. Really, otherwise you get very few ages, very few samples per age, so you don't have enough statistical power. So definitely there's some bias in which predictions we make. They might be actually biased towards brain.
And then you just remove the genes that don't
we remove the genes that don't meet the cutoff and remove all of the interactions. But we keep all edges between the active genes, and there's a way to get rid of some of them. And I definitely think we should be keeping some genes that are not active because they might be critically connective active network parts. So we are aware of lots of room for improvement there. Mm-hmm.
Those are the same patients and different times or different?
No, it's different patients. That's the limitation of why we are not able.
If people in the original data set look at relatives,
Brain samples?
perhaps the bias could be normalized by looking at sibling data.
So these are all post-mortem data that you collect. So we are limited even with collecting data from different individuals. We are very limited what we can collect. Yeah. Hence the need to do something computationally about human aging in general. Okay. So first thing we did is, does global network topology of the age, I will call these age-specific networks, we have 37 of them, do their global network properties change? We did lots of, trust me, this is just three kind of key findings. We did lots of comprehensive analysis, and our general finding is no matter what we did, general topology does not change, which I guess is good. The cell does not completely disconnect or die as we age. But we were wondering what might be happening, so we zoomed into the local level around the genes. And when I say local, I don't mean direct neighborhood. I don't mean even third neighborhood around the gene. I mean whichever property you use to define network position of a node. By local, I mean we are focusing on property of a node within a network. But property of a node can be characterized mathematically virtually via infinite number of options. We use 7 such properties, which are called node centralities, that basically tell you whether a node is peripheral or not, and they are quite different. I will show you how complementary they are. They're different mathematical notions that capture different level of peripherality or centrality. Don't think about what it means for a node to be central in the network in 3D or 2D visual space. Really, this is math we are relying on. So, for each node, at each edge, we compute its centrality for a given centrality value, and then we try to analyze how these time series of the results changes with age. Is there any signal that is interesting or not? Of course, in the process, we compute statistical significance of correlation with age. I'm intentionally using quotes because I don't want to go into more details what we do there. And just to give you an intuition, if this is 3 different networks, at different times or different ages, nodes are positioned exactly where they are in each network in 2D space. If we focus on this particular node, it loses connectivity as captured by degree with age. So that would be an indication that network position of this node changes with age, and we would predict it as aging related. Degree is just, of course, a cartoon illustration of what we do. We use a lot more constraining properties, and we use 7 of them: betweenness, closeness, clustering coefficient, degree, eccentricity, graphlet degree, and k-coordinate centralities. And for each of the centralities, we show you how many of genes we identify whose network position significantly change with age. The one that captures the most genes is the graphlet-based measure that I developed together with Natasha and other collaborators back in 2011. And FYI, at that time, we showed that this particular measure beats a propagation-based measure based on random walks called Estrada index or subgraph centrality. So I know there's been lots of discussions about propagation-based ideas. I think the hypothesis behind it is great. There could be some alternative solutions to the problem. Question?
Very quick question. In the original data, was there any disease with the? I know those people study things like Alzheimer's. And
this was not biased towards any disease, but when we validate the predictions we make, we will go to list of brain-related diseases. To my knowledge, yes, they were healthy, or at least based on the
some diseases that are age-related. So
yeah, I don't think that any of the 2 data sets were biased towards the disease. This is the union of all the predictions, and this particular measure, that happens to be ours, captures almost all of the predictions that are captured by all other centralities. Now, for network scientists, I want to show you an interesting finding without spending too much time on it. This is the number of the predictions that were supported by at least exactly one centrality, exactly 2 centralities, exactly 3, 4, 5, 6, 7. Very few predictions were supported by multiple centralities, which means all of these 7 different mathematical notions are quite complementary to each other. So next time, if you're an applied scientist and you want to just use a centrality measure, think twice which one you choose, because you might get a completely different set of results. And the next question is, okay, we have 5.7 of all genes, 500 something, 37 of them total, that are identified by at least one centrality as aging related in terms of networks. So how do we validate them? In addition to at every step measuring statistical significance of the prediction, we compute statistical significance of the whole approach by randomizing the data, running approach of random data, and making sure we cannot get the same results by chance. And then we do something that is typically done in the bioinformatics community. Can we find some of our predictions in independent data sets that we have not used in the process of making predictions? Do they share functional and disease overlaps with known aging-related genes? And also, are they enriched in aging-related processes and diseases? And finally, for some novel predictions that we really can't find anywhere else, can we find them in the very recent literature and try to validate them that way? I will quickly guide you through some of the results here. These are 4 sets of independent, known ground truth aging-related genes. I'm saying known in quotes because remember they're all predictions from gene expression or sequence analysis. I'm actually lying to you. In the first case, this is the same gene expression data that we used. I don't know if this is visible in terms of color. There's 2 colors here, darker and lighter, darker and lighter, and then this is the percentage of the genes that are in the ground truth data sets, and for 3 out of 4 of them, there's significant overlap. Why am I showing you this? Even though we used it to produce predictions, there's 100+ predictions that can't be captured by gene expression data alone. When we add that data to the network, we can capture novel predictions. We did systematic analysis of Go terms. I want to show you some cherry-picked examples that illustrate the point well. Our predictions are enriched in cell aging, and these are examples of 4 disease ontology terms that are known to be aging-related. For example, known aging-related genes are also enriched in them, and there's articles supporting links of these diseases to aging. 6 diseases were enriched in our predictions. We call them Dynate age, that were missed by any ground-truth data. And then we went to the literature and tried to see whether we can link some of those diseases to aging, and for 5 of them we were able to do so, which is additional encouraging result that our predictions might be complementing aging-related knowledge that is already out there. And finally, when it comes to novel predictions, about 100 of them were not found, 100 of our predictions were not found in any existing at least those that we explored, aging-related data sources. We picked top 10 of them. We have a way of scoring predictions based on how many centralities support a prediction and the p-value of the significance of the correlation with age. And 8 of them we managed to find direct or indirect. For 8 of them, we managed to find direct or indirect link to aging in the literature. Here is paraphrased phrases and the PubMed IDs of the corresponding articles. Very briefly, question?
Do you think we could test these 8 proteins experimentally on?
I am more than happy. All the data is publicly available. I'm happy whoever is doing this experimentally to test it. I'm currently, well, I've been in discussion with Buck Institute for Aging, which is actually here close to San Francisco, of trying to see what we can do with our predictions or take their data, make new predictions, and maybe that they would be more interested that way. So that's the next goal, definitely, to experimentally validate some of these predictions.
These genes are not based on gene expression, right? Their topology is different with the changes with aging.
Yes, but gene expression is a part of the prediction process, but particular gene expression data set. And these are not found in gene expression that we used or any other gene expression data that we explored or sequence-based predictions.
Here's the experimental validation. Knocking out or increasing expression is not...
Well, they might not have sequence homologs in other species, and maybe they are not supported by gene expression, but maybe networks do. That's exactly the hypothesis.
But experimentally, it's hard to investigate network topology.
I'm sure we can dig out some examples that experimentalists would be interested in. Teresa.
Code this last slide. I was just curious. Paul, this is Paul epsilon 2, the last one, the last gene.
This is how it's called in the data. These names really don't mean anything to me as a computational scientist. That's exactly how they're called in HPRD database. Yeah, we provide online unique gene IDs for each of these so that you avoid the bias of translating between different databases. Yeah. Okay, so I will try to give you a quick overview of a novel methodology. The first thing is proof of concept that dynamic analysis actually can be very useful to study human aging. Here we introduce a novel methodology that's established that's based on a very well-established idea of static graphlets. The motivation was we compute property of a node in this subnetwork, in this age-specific network, and in this one, and in this one. And then we study time series of the results. But we really ignore any relationship at the interface between these different networks. So can we define a network property that spans multiple snapshots? In this case, we are dealing with graphlets. I'll show you a little more detailed examples by assigning temporal order of events on top of the static edges that we have. So we are no longer dealing with static graphlets. This 3-node path spans multiple snapshots, and 2 actually, and this 3-node triangle spans 3 snapshots in this example. Just to give you a little bit of formal idea what we are talking about, just the static graphlets, and these are all possible graphlets on up to 4 nodes, are equivalence classes of isomorphic connected subgraphs. Dynamic graphlets, we define them as equivalence classes of isomorphic delta T connected subgraphs, where delta T connected means between every pair of nodes, unordered pair of nodes, there is a delta T time respecting path between the two nodes, and what that means is for any consecutive pair of events along the path, such as Monday and Tuesday, Tuesday and Wednesday, or if I had Friday and Saturday, or Saturday and Sunday, there can be at most delta T time between those 2 events before they are considered delta t adjacent. So there's lots of formal definitions we introduced in the paper, but bottom line is in both of these examples, Monday, Tuesday, Wednesday, Friday, Saturday, Sunday, if my delta t is 1 day, this will be my resulting dynamic graphlet. Again, 1, 2, 3 corresponds to temporal order of edges in which they occur. If I had Monday, Tuesday, Thursday, what does my delta t need to be so I have 1, 2, and 3? Because Tuesday and Thursday are 2 days apart, my delta t now would have to be 2 days. If I had Monday, Tuesday, Wednesday, Thursday, no problem. We allow multiple events over the same edge. And here are, this is the same example I already showed you. In the interest of time, I'm just going to speed it up. This is the general idea. Here are all possible dynamic graphlets on up to 3 events. We order them based on number of events, and in this case, up to 4 nodes. Okay, when we do computations in graphs, we go up to 6 nodes and up to 7 events. And you will see that there's multiple dynamic graphlets that correspond to the same static graphlet if you ignore the temporal dimension on the edges. So we had to deal with lots of not problems, research questions. How many graphlets are there with n nodes and k events? Dynamic graphlets. We first derived a recursive formula and then found the closed form solution. And then once you know what you need to count, how do we actually count it in the network? We have 2 ways of doing it. I'm not going to go into details in the interest of time, but the output of this methodology is either for the whole network, for the whole dynamic network, how many graphlets of each type, how many dynamic graphlets of each type appear in the network, or for each node, how many dynamic graphlets a node participates in. This is the concept of graphlet degree vector of a node with respect to dynamic graphlets, and we do take into account automorphism orbits when we do this. I'm going to show you 10% of the evaluation part of this paper and be very close to concluding my talk. When we used, if you consider static PPI network and try to use graphlets to make predictions, we are calling those static graphlets. What we've done before, because we are using static graphlets in each particular snapshot without any relationship between snapshots, we are referring to that analysis as static temporal graphlets. And dynamic graphlets are those that span multiple snapshots at once. And we are here trying to hide the knowledge about the given known aging-related gene and measuring how accurate we are in predicting it. So I'm showing you kind of precision equivalent of this analysis. You can see statistics for random predictions. Static already is better than random order of magnitude. Even static temporal, look how much it improves compared to static analysis. So next time you do static analysis of the cell, think how much you can improve doing something dynamically. Although it's not fair, these two are not fair to static only because they do include gene expression data, which static does not. And then dynamic actually improves drastically. Again, it might not seem drastic, it's statistically significant improvement. And when we try to go to novel predictions by each approach and try to validate them against sequence-based predictions, so this is equivalent of GenAge, the most trusted source of aging-related data in human out there, only dynamic graphlets produce statistically significant functional and disease overlap with the known genes. In the interest of time, I'm going to skip examples of Go and terms and those terms that we found that are novel to dynamic graphlet-based predictions that we managed to validate in the literature. And this is overall comparison of the different types of graphlet approaches where dynamic are superior in 2/3 of all cases and comparable in the rest. So static analysis is definitely, let me start with this, and dynamic analysis is definitely better than static temporal, which is definitely better than static. And we can use dynamic graphlets to characterize the structure of the entire network or individual nodes. Static graphlets have been used in very many computational and applied tasks. There's no reason why you can't do the same with dynamic graphlets. Code is available online. I'll take 30 seconds to tell you some key comments on network alignment without telling you what we do or how. We have a new method for pairwise network alignment. Magna is definitely the methodology, but we improved it with lots of other features, user-friendly graphical interface into Magna++, and recently we extended this to multiple network alignment, and the new paper is on archive, although not published yet. Global network alignment in how we used it for the purpose of aging. I want to point anybody who works on the topic of network alignment, especially global one, read this paper. Even today I'm reading papers and they're not fairly evaluating their method against existing ones for a very simple reason. But definitely please read, and this is our recent survey on the topic. I already acknowledged most of my group. Just let me mention that Fazl was the one working on the first study of studying dynamic aging through dynamic networks, and then the topic of dynamic graphlets was done by Yuri and Huili. And NSF, Air Force, Google, and NIH are the groups or agencies that are funding my group. I'll be happy to take any questions. Thank you.
Can you speculate on this underlying mechanism, like just take a gene and its centrality is changing with age? What is going on that can be decoupled from changes in gene expression alone?
We've been asked this question. The short version is I don't know. Long version is networks might be capturing different things than expression data. It could be that networks, especially the measures we use, go even beyond the neighbors to their neighbors, and graphlets go like depth 4 around the given node. I'm not going to go into details why, and all around it, and given small world nature of real world networks that might be capturing the entire network. So it's simply capturing indirect relationships. I don't know, but I have some ideas, but I really can't even, they might be completely wrong, so I would prefer not to discuss in detail. I'm just going to say power of networks.
Have you compared your methods of the dynamic graphlets with Westheim's generalizations of paths and cycles to dynamic versions?
We have not. We have seen lots of work in social network research about dynamic motifs, but they are defining dynamic motifs with respect to random graph models, so we were not aware of that work, and yeah, so the answer is no, but definitely something we will take a look into.
So in the dynamic graphlets, you have this time concept, but is it essential to have like really time? Can you use the same idea for, say, different tissue types, which is like a spatial dimension?
You are adding with labels on edges as we currently have. We know the order of labels.
So it's important.
It's extremely important, because if you don't know the order of labels and you increase the number of labels, you will get exponentially huge number of graphlets that you can't even deal with. So that's a completely different problem. People have tried to address it, and with 4 labels on up to, I don't know, 5, there's millions of options. So there's way too many options. So it's a great question, and I would love to come up with a way to solve the problem that way, but it's not directly applicable.
Let's thank Tijana again.
Thank you.